Make sure you go through the additional points about glycolysis given at the end of this video to develop a comprehensive understanding of the entire glycolytic pathway. The first step in glycolysis involves the action of the enzyme hexokinase on glucose. Any enzyme that has a kinase in its name can be called a phosphotransferase, as it adds a phosphate group to its substrate. And it acquires this phosphate group most often by the hydrolysis of an existing ATP molecule to ATP. That's why the product is glucose 6-phosphate. This step, being irreversible, traps the glucose molecule inside the cell, as glucose 6-phosphate, being charged or polar with the charge conferred by the phosphate group, cannot easily escape the cell. But glucose, in spite of being hydrophilic or polar, can pass through the hydrophobic core of the cell membrane. And that's because of a carrier protein called glucose transporter or GLUT embedded in the cell membrane. It was a GLUT that brought glucose into the cell to begin with. Glucose 6-phosphate then undergoes isomerization to form fructose 6-phosphate by the action of phosphoglucoisomerase or phosphohexose isomerase. The next step is the rate limiting step, in which fructose 6-phosphate is phosphorylated by a kinase called phosphofructokinase to fructose 1,6-bisphosphate, with the phosphate coming from another ATP molecule. This is the second irreversible step. The third and last irreversible step is the last and 10th step of glycolysis itself, as we shall see later. In the next step, 6-carbon fructose 1,6-bisphosphate is cleaved into 2 3-carbon molecules by the enzyme aldolase. One of these molecules is dihydroxyacetone phosphate. But dihydroxyacetone phosphate cannot be metabolized to generate ATP. That's why it's isomerized to a more usable form, glyceraldehyde 3-phosphate, which is the other 3-carbon molecule. So we have 2 molecules of glyceraldehyde 3-phosphate which are broken down simultaneously in 2 parallel reactions yielding the same product. That's why every molecule in all of the subsequent reactions is multiplied by 2. Additionally, since both dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are triose phosphates, the enzyme catalyzing the isomerization reaction is called triose phosphate isomerase. You can notice that every molecule has a coefficient of 2. This is true for every reaction that follows. Now the next key enzyme is glyceraldehyde 3-phosphate dehydrogenase. As the name suggests, it removes the hydride ion from glyceraldehyde 3-phosphate and adds it to an electron carrier called nicotinamide adenine dinucleotide or NAD+, present at its active site, and reduces it to NADH. After this, inorganic phosphate present in the cell as anions gets added to glyceraldehyde 3-phosphate, converting it into 1,3-bisphosphoglyceric acid and releasing a proton in the cytosol. Additionally, since a hydride ion has 2 electrons, NADH has 2 electrons more than NAD+, which it carries to and releases in the electron transport system in the mitochondria, as we shall see in a subsequent video. Phosphoglycerate kinase then transfers a phosphate group from 1,3-bisphosphoglyceric acid to ADP, forming 3-phosphoglyceric acid and a molecule of ATP. Since this ATP is formed directly from the substrate, this step is called substrate level phosphorylation. 3-phosphoglyceric acid is then acted upon by phosphoglycerate mutase, which forms 2-phosphoglyceric acid. A mutase is an enzyme that facilitates intramolecular phosphate transfer, and an enolase is an enzyme that causes dehydration. That's why removal of a water molecule from 2-phosphoglyceric acid yields phosphoenol pyruvate. So we have no more glycolysis beyond this step, and as I promised you, this would be an irreversible reaction, so it is. And there are kinases galore. Pyruvate kinase transfers a phosphate group from phosphoenol pyruvate to ADP, forming pyruvic acid and a molecule of ATP. For reasons cited for a previous reaction, this is the second example of substrate level phosphorylation. So by the time fructose 1,6-bisphosphate was formed, 2 molecules of ATP had been consumed, which was recouped in the first substrate level phosphorylation. And now we have 2 as surplus. So the net gain of ATP in glycolysis is 2 molecules. Now both the molecules of pyruvic acid entered the mitochondria to initiate 2 rounds of the Krebs cycle. Glycolysis may seem to be complete, but I have some more facts to convince you otherwise. Hexokinase was the enzyme that catalyzed the conversion of glucose to glucose 6-phosphate in the first step. Another enzyme that could have catalyzed the same reaction is glucokinase. Glucokinase has a much lower affinity for glucose than hexokinase. That's why it's activated only postprandially, that is after a meal, when the blood glucose level is high. It also acts as a glucose sensor, triggering the release of insulin from the beta cells of the pancreas and promoting glycogenesis in the liver. That's why it's primarily found in the liver and the pancreas. On the other hand, hexokinase is ubiquitously found and constitutively expressed, even in the fasted state. Constitutive expression means it has a certain basal level of expression at all times necessary for survival. It's always switched on and is not subject to regulation. That's why it's also called a housekeeping enzyme. In fact, all of the glycolytic enzymes are called housekeeping enzymes. And amongst them, glyceraldehyde 3-phosphate dehydrogenase or GAPDH is the one which you will encounter frequently. These enzymes also have moonlight functions, which means they perform other activities beyond their primary role. You can refer to the English meaning of moonlight for a clearer understanding of the term. Glucose 6-phosphate is a metabolite of galactose, permitting galactose to enter the glycolytic pathway. Similarly, glyceraldehyde 3-phosphate is a metabolite of fructose, permitting fructose to enter the glycolytic pathway. All of the 3 irreversible steps are the slowest, but the pacemaker of glycolysis catalyzes the rate-limiting step. The pacemaker enzyme, that is phosphofructokinase, is more precisely called phosphofructokinase 1. The reason being the existence of another enzyme called phosphofructokinase 2, which converts fructose 6-phosphate to fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate in turn upregulates phosphofructokinase 1. You must have heard about the oxygen hemoglobin dissociation curve, and that there are some factors which make it shift to the left or right. But have you heard of 2,3-bisphosphoglyceric acid? Don't get confused with 1,3-bisphosphoglyceric acid which we found in glycolysis. But if a mutase acts on 1,3-bisphosphoglyceric acid, it forms 2,3-bisphosphoglyceric acid, which shifts the oxygen hemoglobin dissociation curve to the right. So that's it for glycolysis.